mechanism. Its mechanism for transduction of hormonal signal from cell membrane receptor. So one more time. This is the cellular membrane. I remind you that phospholipids, they are component of membrane. Two tails, one head. So heads are hydrophilic, tails are hydrophobic. On this cell membrane, we have a receptor. So this is receptor. When hormone connected with receptor, receptor change the conformation and it starts to act with G protein. I remind you that there are three subunits in structure of G protein: alpha, beta, gamma. Alpha subunit connected with GDP in case of hormone absence. When hormone connect with receptor, receptor start to connect with G protein, GDP will go away, and at the place of GDP, we connect with GTP. Alpha subunit with GTP activate hormone in our membrane. It's called phospholipase C. Phospholipase C. Phospholipase, this is hormone what hydrolyzes our phospholipids. So in structure of our membrane, we have phosphatidylinositol biphosphate. This is the name of one of the phospholipids. Phosphatidylinositol biphosphate. Phospholipase C will take this phospholipid and separate it for two structures. From this lipid we can get inositol 3-phosphate. This is a part of head of phospholipid, hydrophilic part. And we have still more fatty acids and component of the salt glycerol part. So it will be diglycerol. So this is a piece of head, and here is another piece of head and two tails. Inositol 3-phosphate will connect with endoplasmatic reticulum. And we know that endoplasmatic reticulum, this is an intracellular store of calcium. So inositol 3-phosphate will activate calcium channel and calcium level increase into the cell. The first action is movement. So if this cell can contract, we will provide muscle contraction, for example. But also calcium will regulate metabolic weight. You know, it will increase the rate of the Krebs cycle, for example. Calcium will regulate transcription, transduction. For example, how we can regulate gene expression. Calcium will connect with specific calcium binding protein, calmodulin. And can regulate even gene expression if we will transfer it into the nucleus. So this is the one side. Yeah, in that case, calcium works as a secondary messenger. And other side, diacylglycerol will activate protein kinase C. Protein kinase C, this is an enzyme which will provide phosphorylation reaction. So again, we will regulate metabolic reaction. We can regulate some mechanisms of hormones, enzymes activation. And last way, what we can do in this diacylglycerol, if we will separate some fatty acids from structure of diacylglycerol, we can get eicosanoids. This is a prostaglandin, prostacyclins, leukotrienes, and thromboxans. So it can be an answer of the cells if we will activate some fatty acids regulation. So this is a cell membrane transduction of hormonal signal. So please take a photo of it. We have it in our books. So this is a very often asking question. The second task was T3, T4 synthesis. T3 is triiodothyronine. T4 is tetraiodothyronine. Another name is thyroxine.
Professor, I like this 3535.
I will check it later. So, we talked about thyroid hormones. Thyroid hormones synthesized by our thyroid gland as the action of thyroid stimulating hormone of hypothalamus. So I remind you that thyroid gland contains specific structure. We have a folliculars in the thyroid gland. So what is this folliculars? We have some kind of bubble.
We studied in histology. We studied this in histology.
Yes, I just remind you.
Oh, okay.
We have a bubble with a specific liquid, it's called colloid, and it's formed by follicular cells. So we start synthesis of our T3, T4 in follicular cells of thyroid gland. Now let's increase our picture. So it will be follicular cell. And it will be colloid. Inside of the bubble, liquid here. So in follicular cell, we have ribosomes, and on ribosomes, we will synthesize specific protein. It's called thyroglobulin. On ribosomes, we will synthesize thyroglobulin. What do we know about structure of thyroglobulin? It is rich by tyrosine amino acid. So there are a lot of tyrosine residues on the structure of thyroglobulin. It will be more than 100 tyrosine residues. Correct number of 115 tyrosine residues in thyroglobulin structure. So thyroglobulin will be packed into the vesicles, and these vesicles will be excreted from the cell into the colloid. So we have thyroglobulin in colloid. So this is a protein, this is a protein. So first what we will do in colloid, it will be iodination. In this case, we have a very specific hormone. It's called thyroid peroxidase. What do we need to do? We need to oxidize iodine in colloid and transfer it in the structure of tyrosine. So we take iodine plus as the oxidational molecule, we will use hydrogen peroxide. That's why this hormone name peroxidase. And provide formation of residues of tyrosine with iodine. So it is understandable that we can get DIT, diiodothyronine, and
MIT.
MIT, monoidotyramine. So if we will add 2 iodine in tyrosine, it will be dietine. If we add only 1, we will get MIT. So next step, it will be condensation. So we will transfer residues of tyrosine into the other residues of tyrosine. Something like this. Wait, minus, everywhere minus. So if we will condensate DIT with DIT, it will be the precursor of T4. If we will connect DIT with MIT, it will be the precursor of T3. But it's still on the structure of tyrioglobulin. Do you see we don't cut protein here. So still here we have a protein part. After it, by exocytosis, so we form a bubble here. Put this protein into the bubble. We transfer it again in the follicular cell of our thyroid gland. So this bubble and the endosome connect with lysosome. Do you remember that lysosome is the organoid which contains a lot of hydrolytic enzymes? And these hydrolytic enzymes will cut protein part and free our hormones. So T3 or T4. After it, we will release these T3 and T4 into the bloodstream. So it was colloid, it was cell, and this is blood. I remind you that T3 and T4, they are hydrophobic. So we have a transport protein for transport of these hormones. So some kind of protein connect with our T3. T3 is more active than T4, but amount of T4 is higher than T3. T4 is more stable also in our blood. So this is the synthesis of thyroid gland. Thyroid hormones in thyroid gland. And third question was clinical cases. Some of the students asked me the same questions. So I underlined it right now by common discussion. If you don't answer to my question, what example do you want? I answer for another question, metabolism of calcium. So, previous class we already started discussion about insulin, and you know that insulin is a simple protein. It synthesized in beta cells of pancreas. It synthesized in form of proinsulin. After it, we cut C-peptide, connect A and B part with disulfide bond, and we have an equimolar concentration of insulin and C-peptide in our organism. Insulin is active. Insulin will be very fast destroyed by our nuclei, our proteases in blood. But C-peptide is more stable, so we will prefer to use it for counting of insulin concentration. And also, I remind you, this is a way of the counting of insulin level in patients who already take insulin as a drug. Because in this case, we take only insulin, and C-peptide is show our own level of insulin. So, a lot of students ask me about tests for diabetes mellitus diagnostics. So, in the question, there are two ways of diagnostic: bedside test and laboratory test. Bedside test and laboratory test. you are not laboratory doctors. So I'm laboratory doctors, I work in laboratory in Children's Hospital. So you will not take a blood sample, do something with it and get a result. But sometimes in department of endocrinology, we have some very cheap reagents, very cheap equipment to check right now some analysis. So first of all, it will be glucose in blood. A patient can have their own glucometers, or in department we have a portative glucometers. So we take a drop of blood, by finger capillary blood, put it into the it's some electron equipment, put it into the glucometer and get a level of glucose blood. So it's not very, it can be some mistakes for counting of blood level, but we can count that it's increased, high or decreased. So somewhere between 3.3, 6.1 millimole per liter. So this is the blood glucose level. What else we can do in endocrinology department? This is a urine test. We have a urine sticks. So we will put into the urine, wait for some seconds, and check colors with stick and with pictures on the tip of this stick. So it will be Benedict test, glucose in urine. The stick is green. When we put it into the urine where we have a glucose, green color convert to yellow color convert to green. If we don't have any glucose in urine, we have still yellow color. And Rotera test. It was also written in some clinical cases. This is ketones in urine. Colorless or white stick we will put into the urine. If urine contain ketones bodies, it's become purple or pink. So again, this is a paper sticks with some reagents, what we put into the urine, and very fast, after 5 seconds, we can count and check the color of this stick. So what we can do in bedside, so in endocrinology department. What you will do, what I will do in my laboratory. So first of all, yes, of course, I will check glucose in blood. But now I will take the venous blood and will separate it to the blood serum or blood plasma, even plasma. You count it into the whole blood, in the capillary blood. I will count it into the blood plasma. Let's write same. What I will do next? I will count insulin level and C-peptide level. This is immunochemiluminescence analysis I will do by the analyzer. So we know insulin level very low, this is a diabetes mellitus type 1. C-peptide very low, diabetes mellitus type 1. But I need to check if this patient get insulin as an injection. In that case, it is possible that insulin normal, C-peptide low. So insulin, this is that insulin what patient take with injection. What I will do next, I will count in whole blood glycosylated hemoglobin. Now you already know how we write it in laboratory. Hemoglobin A1C. There are three types of glycosylated hemoglobin, A, B, C. For diagnostic, we will check one more stable glycosylated form, and this is a hemoglobin A1C. A1 is adult hemoglobin. A1C is glycosylated adult hemoglobin. How it's happened? In diabetes patient, glucose level very high. And you know that glucose can make glycosylation without any enzymes. So it will connect with proteins, connect with membranes. So glycosylated hemoglobin show us the glucose level of this patient in previous 4 months. Because I remind you that hemoglobin is the main protein of RBC. And RBC period of life is 120 days. So glucose blood can show us glucose level right now. Glycosylated hemoglobin show us if we have hyperglycemia on previous 4 months. So we will use this test each 3 months of patient with diabetes mellitus and understand if our treatment is correct. So is glycosylated hemoglobin level in normal condition? We understand that glucose will be average of the normal way, and we make a correct treatment. You must understand that diabetic patient will never have a normal range. For example, healthy persons will have glycosylated hemoglobin no higher than 6%. For diabetes patient, target amount will be 8%. So it will be higher than in normal persons, but it will be still somewhere near the norm. So target concentration of glycosylated hemoglobin for diabetic patient is 8%. What else we can do in laboratory? Do you remember that diabetes mellitus type 1 is autoimmune disease? So very often it's happened in childhood, and our immune system start to destroy beta cells of pancreas. So what antibodies we can check? Anti-gut. So this is a glutamate decarboxylase. This enzyme present in beta cells, so patients with diabetes mellitus type 1 will produce antibodies through this against this enzyme. What else? ICA, islet cells antibody. This is a common antibody against beta cells. It's called tyrosine phosphatase. These antibody. Again, tyrosine phosphatase, this is enzyme involved in insulin secretion. Against this enzyme, we can have antibody in our organism. And last, what we use? Anti-zinc TA. Zinc transporter. Number 8. Zinc involved in stabilization of insulin in the vesicles. So again, patients can produce antibodies against the transporter of zinc in beta cells. So very fast you check glucose level, very fast you check hormones level. We can check what's happened on previous 4 months, and we need to confirm diagnosis. So if we see that this is diabetes mellitus type 1, we need to find any of antibodies in the patients. If we don't have it, it will be another type of diabetes. It can be monogenic diabetes, inheritance type of diabetes, or diabetes mellitus type 2. Next class, we still have new topic, metabolism of calcium phosphate level. So regulation of the calcium level in blood and in salivary. Now this is your topic about dentistry diseases of the hypercalciumia, hyperphosphatemia, and corruption of teeth. I just start to check your tests. I will finish it later. I will check all your written tasks today. Now you know what you need to, what you must write for me. Next class, we will see maybe some short clinical, maybe some short written tasks. Maybe I will ask you orally. So that's all. You're free. Goodbye.
نمیشه این هفته هم هایر کنیم برای بیوشیم